Can you see the screen? Right. So welcome to the University of Connecticut's Brain Imaging Research Center, or BIRC Speaker Series. I am the host, Fumiko Haft, Director of BIRC and Professor of Psychological Sciences, Math, Neuroscience, and Psychiatry. I apologize, I'm admitting people as I speak, so I will be a little distracted, but I apologize for that. Over the past 2 years, we've hosted a number of speakers, as you can see here, most of whom their recordings are available on our website. We've had about 2,500 or maybe 3,000 now people joining our series from over 150 institutions all over the world. Thank you so much for helping us make this event so successful. Please do let your friends and colleagues know about the series, and they can get updates by emailing Lizzie, that is listed here under listserv. So some housekeeping items that you probably all know by now. You'll be muted, and we usually try unmuting. I know everyone's laughing probably because every time I fail, but I'm going to try again today. I like unmuting so that people can clap before and after the talk, as we feel like it's always nice and gives a little bit of a human touch to this. And even though it's a disaster, but we'll give it another try. If you have questions, please enter it in the chat box, and we will take most questions at the end. So let's get started. Today, it is my greatest pleasure to introduce Professor Allyson Mackey, who's an assistant professor of psychology at the University of Pennsylvania and is affiliated with the Neuroscience Graduate Group, the Center for Neuroscience and Society, and the Positive Psychology Center. Allyson received her bachelor's degree from Stanford University in biological sciences, then did her PhD in neuroscience with Professor Sylvia Bungay at UC Berkeley. I see that she's here, followed by a postdoc with Professor John Gabrielli at MIT. So we are like distant second siblings or half siblings, maybe, because I did my postdoc with John Gabrielli as well. And she has been assistant professor since 2016. I met Allyson when she was an undergrad at Stanford University, and she came to our center to do some neuroimaging research. I think it was in 2006 or so, quite a while ago. And I am honored to have been co-authored her first neuroimaging paper. So I saw her as a rising star back then in 2006. So, and she kept her, she didn't let me down. She's now a CFAR, I apologize, I don't know how to pronounce it properly, but Azrieli Glober Scholar, a Jacobs Foundation Scholar, and serves on the Scientific Advisory Council of the National Center on the Developing Adolescent. She is a recipient of the Rising Star Award from the Association of Psychological Sciences. Allyson directs the Changing Brain Lab. She studies individual differences in brain plasticity and development with an eye towards personalizing the type and timing of educational interventions. Her research is supported by the National Institute on Drug Abuse, the National Science Foundation, CFAR, and the Jacobs Foundation. So excited to hear from Allyson today after many years of not being in touch. And please join me in welcoming Professor Allyson Mackey. So I'm going to try to unmute everyone. Let's see. I do not know how to do this. I am not seeing the button here, so I am going to give up, and I'm going to be the only person clapping today. But thank you very much for coming, and I'll give the screen back to you.
Thank you, Fumiko. I think that you have undersold your role in my career. You taught me to love cognitive neuroscience, how to do it, and just how wonderful and cool it could be. So thank you for that, for launching my career and for inviting me today. But before I fell in love with cognitive neuroscience, my first passion, the first thing I deeply cared about was understanding inequalities in the classroom, inequalities in how children learn. And this interest arose from being in these kinds of classrooms in severely underfunded urban schools. And I'm feeling through a lot of these experiences, remembering them more as I read about Philadelphia's efforts to reopen schools during COVID, how none of these schools have hot running water, how they don't have soap, right? That was exactly my experience as a kid in these schools. And it can be so frustrating to see how little progress we've made in closing these gaps, and especially seeing how much bigger they're getting as kids have not been able to learn during the pandemic. But what I hope is that this severe disruption will actually become an opportunity, where we can sort of revisit what school's for and bring in new science into designing the classrooms of the future where kids can learn better and to thrive. And I think there will be a role for cognitive neuroscience in this future. And of course, the way we think about these issues from a cognitive neuroscience lens is trying to understand how the brain gives rise to the mind, gives rise to the processes kids need to succeed in the classroom, not just their math and their reading, but also domain general skills like reasoning or creativity, curiosity. And there's more and more interest in understanding how the environment shapes the brain. And a lot of what we know about these relationships comes from correlational cross-sectional evidence. And we've learned so much through these methods. But what we really strive for is to understand the deltas better, to understand how changes in the environment give rise to changes in the brain and changes in the mind. And we can do that in a number of ways. We can root our hypotheses in causal animal models, right? So we can understand directly how these changes change the brain. We can focus more on longitudinal studies where we can establish temporal precedents, right? Changes in the environment happening before changes in the brain. And we can actually go in and change kids' environments, right? Both in the short term by borrowing methods from experiments in developmental psychology and in the longer term. So following kids as they experience more intensive holistic interventions with sort of greater likelihood for enduring change. But the key to understanding this whole framework is understanding these changes in the brain or brain plasticity. And so much of what we know about brain plasticity was discovered at the level of cells and synapses, right? So we know that cells that fire together, wire together. This is the process of change. We see formation or strengthening of synapses. We see formation of myelination, changes in glia, such as astrocytes that can change much more quickly than neurons. But another way to think about plasticity is to think about it as the ability to change. So here, when these cells fire together and wire together over time, this formation of myelination not only makes the system more efficient, right, it also glues it into place, right? So you can also think about this over the course of a child's development. As they grow up, more and more myelin gets put into place, right? Preventing future changes. And we see that too with inhibition. So we know that the maturation of these parvalbumin positive interneurons locks down on plasticity. And you can think about it in terms of how inhibition is changing the role of this neuron to make it more different from this neuron. So this was discovered in ocular dominance columns in visual cortex, but the same processes might also be at play if we think about the landscape of prefrontal cortex, regions of prefrontal cortex caring more about the internal world versus the external world, and inhibition creating those kinds of separations over time. We also know that over development, there's the formation of perineuronal nets, these extracellular matrix proteins that sort of glue into place a particular pattern of synapses. So collectively, you can think about these processes as brakes on plasticity, right? But the brain is actually pretty good at regulating when to let up on these brakes, right? So there are systems in place to reawaken juvenile-like plasticity in adult animals. So there was a landmark study by Mike Merzenich and Xiaowen Bao showing that stimulation of the ventral tegmental area, which creates dopamine, actually leads to juvenile-like plasticity in auditory cortex in adults. One of my very favorite studies looked at barn owls. And what they did, this is very cute in my mind. They put prism glasses on baby barn owls to misalign their auditory world from their visual world. And in baby barn owls, the brain just rewires. Right? Easy peasy, as my daughter says. In adult owls, the networks sort of stayed misaligned, right? Until the adult barn owls had to hunt, right? When there was motivation in play, right? Their brains rewired just like juveniles. And we're learning more and more about sort of the dynamic processes to regulate plasticity. So there was a study that I love that came out last year showing that in the presence of cognitive enrichment, right? So new social interactions, new toys. In mice, what you see is that glia, microglia in particular, are activated to actually eat up these perineuronal nets, reawakening plasticity, right? So we know that cognitive enrichment can causally increase plasticity. But because you're all cognitive neuroscientists, you'll be thinking about just how small these cells are, right? Everything I'm telling you about takes place on the order of 1/10 of a millimeter. And when we compare that to sort of best case scenario resolution with neuroimaging, we see that voxels look really, really big, right? And if we're thinking about functional neuroimaging, it's 2 or 3 times this. But what I like to argue is that voxels are big, but brains are a lot bigger, right? So here's a 1 millimeter voxel for scale across the entirety of the brain. And there's more and more evidence that brains are organized in these sort of widespread distributed networks. And so understanding just a 1 millimeter piece of this is not going to give us the whole story. It's not going to allow us to understand how all of cortex is changing in terms of its development and how development is restricting plasticity. And so I think there's going to be a role here for neuroimaging. And it's going to involve using some proxy measures. Right? And just to walk you through what this might look like, you generally think about neuroimaging as falling into categories of structure and function, of local measures versus connectivity. When we think about local structure, we can think about cortical thickness, so the distance between the outside of the brain to the gray-white boundary. And that's been linked now directly to myelination. Right? So there may be some reflection of synaptic remodeling, but it looks like a lot of it's driven by myelination, pushing out that gray matter signal. We can also think about myelination by looking at tracks. And so people use diffusion here, looking at how water diffuses along a tract as compared to the orthogonal directions. I'll be talking about this less. And when we go to the functional world, so we have most commonly task-based fMRI. A lot of my work today will be focused instead on resting state fMRI, so looking at the correlation in blood oxygen signals between 2 areas. And you'll see that this can be used to map out anatomical connections, but also to look at these sort of broader network dynamics, how areas change in terms of how different they are from neighboring cortex. Okay, so we can combine some of this evidence from neuroimaging with what we know about perineuronal net formation, myelination, to create some sort of model in terms of how plasticity is changing over development. And so we say that development is basically decreasing plasticity, but this happens in different areas at different times. So we think that visual cortex is going to develop first, followed by somatomotor areas, followed by association cortex. I'm just putting frontoparietal cortex here as an example of a late developing set of association cortex. And I think actually figuring out this model is really important, right? Because it tells us something about when plasticity is going to change, right? So if you wanted to intervene to change visual function, for example, you might not want to do it here when there's just very high plasticity, and certainly not here when plasticity is much lower, but you might want to do it here so that anything you're able to change then gets glued into place. And so there's some value in establishing when these inflection points are. There's also value in knowing what the rank order of these systems might be in adulthood, right? We're not going to get to 0. That tells us about what kinds of things we can still change in adults. But really, for this to be helpful, we have to sort of go beyond this sketch of a model to really labeling these axes in detail. Let me just show you one pass at this. And this was done by my graduate student, Ursula Tooley, looking at networks. So here, she compared the adult networks. So I just showed you these same rainbow networks by Thomas Yeo and colleagues. Now I'm showing you all 4 surfaces, so lateral and medial. What she wanted to know is that if you take a snapshot at age 9 or 10, at the ABCD age range at time 1, what do these networks look like? Right, from this model, you would expect visual areas to be mostly developed, maybe motor areas are still changing, and association cortex is still highly plastic. And I think when you look at what she found, you see that kind of pattern. Visual areas look just like adults. Motor areas on the medial surface do too, but if you look at the lateral surface and you look at the ventral aspects, the areas that are responsible for the hands and for the face, right, so potentially important for language and expression, those areas are still linked into the salience network or ventral attention network. And if you look at the landscape of lateral prefrontal cortex, for example, it doesn't look exactly like adults. And what was most striking for us is that we see this limbic expansion, right? So kids have a lot more cortex that's limbic than do adults. And for me, this really links into what kids are like, right? Emotions are highly salient. Their emotion regulation is not adult-like by age 10, right? And maybe that's mapped into sort of what is important in their brains. Another way of asking this question about plasticity is to, instead of using this community detection approach that really focuses on the strongest connections, to use instead a weighted stochastic block model approach that allows you to take in all of the correlations. And there you see the association cortex is just really hard to pin down, right? It doesn't like to be assigned to one network in the same way that some of these other networks are happy being visual or somatomotor. And that tells us that a lot of these boundaries are not yet set by age 10. And this is an approach to trying to understand just typical development of these processes. But what I'm most excited about is really understanding the individual differences. So understanding the kinds of experiences that drive kids to grow up too fast, to go through these changes and lose plasticity in networks before their peers. Right? And what we might do, given that these changes happen earlier, to still support their learning, to be able to drag these plasticity curves back out, kind of figure out if it should be left or right, to my right, so that we can sort of reinstate juvenile-like plasticity or help kids learn even if these breaks are in place. So the questions that I'm going to be addressing into the talk today is, first, let's look at how children's experiences might be impacting the rate of their maturation, both of the brain and the body. I want to think about how children's experiences are related to the development of their dopamine circuitry, because I think this has, again, implications for their plasticity. And then I want to think about what we might need to do to build interventions to harness what we know about dopamine and motivation to help kids learn better, even in the face of accelerated maturation. So let's start with this first question. What I want to do is think about what we should expect in terms of which experiences shift windows of plasticity. And we can create a map from negative experiences to positive experiences, where we know that negative experiences, many of them will initiate stress cascades that will cause allostatic load, wear and tear on the system. We know from animal models that these kinds of stressors decrease plasticity across sort of all of the measures I was telling you about. We know that cognitive enrichment, on the other hand, can enhance plasticity. So these positive experiences might enhance plasticity. But the brain is not going to pay attention just to valence. It's also going to pay attention to timing. So it's going to pay attention to whether these experiences are repetitive or rare. Experiences that are repetitive are going to cause cells to fire together and wire together, going to put in place this experience-dependent myelination. Right? And you can think that this makes sense. If the world is predictable and things keep happening over and over again, then it might be safe to glue things into place, right? Your brain has successfully matched the environment. If instead you have rare experiences, right, that might be a signal to keep this plasticity open, right? That you are not successfully predicting everything that will happen to you. And it's better to take the gamble on maybe high risk plasticity to have the chance to rewire in response to future experiences. And so what I think falls out of this is that we should predict that experiences that are both repetitive and negative will promote breaks on plasticity, while experiences that are rare and positive instead might boost plasticity. And just to give you more concrete intuitions about this, we can tile this with some examples. So we can think about boredom or adverse childhood experiences as something that might be both negative and repetitive. Right? A lot of the ACEs that have been so tightly linked to health outcomes across all systems are very chronic, right? So you're experiencing parents with mental illness or substance abuse or incarceration or going through divorce or separation for long periods of time. We could contrast that with some things that might trigger surprise or awe, right? And much less is known about this positive quadrant. We know way less about how positive and rare experiences are going to affect the brain. But my guess is that they're going to promote plasticity. And here's just a moment to pause and think about what kids' experiences are like right now during the pandemic, right? For some, they'll have many of these negative and repetitive experiences. For many, they'll be missing these positive and rare experiences, right? Kids can't see other people, they can't travel. And so we can sort of generate some predictions about what is going to happen after all of this. To contrast that, we can think about the other quadrants, so positive and repetitive, or negative and rare. And these are the kinds of experiences that might impact specific systems, but might not sort of shift the entire pace of brain development. And what I would like to do ultimately is study these experiences sort of one at a time, because I think that's going to give us more leverage for intervention. But in American society, what we see is that these experiences are correlated. So children from lower socioeconomic backgrounds are going to experience more of these. There's less data on this, but it's also possible that they experience fewer of these kinds of experiences. And so what I'm going to talk about for the next section is really a focus on understanding links between socioeconomic status and the pace of development. With this as a theoretical framework, but there's less direct evidence for these kinds of experiences. So let's first start with looking at links between socioeconomic status and structural brain development. I'm going to show you first data from adolescents. So these are 12 to 16-year-olds. And what we see is that kids from higher income backgrounds, so kids who don't receive free lunch at school, have thicker cortex than kids from lower income backgrounds. And what I expected when I first did this analysis was to only see differences in areas that were specifically sensitive to stress or to language. And what I saw instead were differences in visual cortex, differences in somatosensory cortex, right? Really localized to the posterior areas of the brain, which made me think that what we were seeing was a global acceleration of thinning and not something that was specific to experiences, certain experiences. And we saw that cortical thickness was important for these kids. So kids who had thicker cortex in these posterior areas did better in school. They did better on standardized tests of math and reading, even controlling for their income. And so what I wanted to know next was whether we see the same patterns if we look younger. And so we are going to look now at 4 to 7-year-olds. And what we first want to know is what areas are actually thinning when we look at early childhood. So where are the areas where we see a negative correlation with age? So first, let me show you data that I collected as a postdoc in Boston, where we see it just in V1. This is the medial surface of the brain. If we look in our Philly sample, we see the same features in a visual cortex, and we actually see broader areas too. But I will note that our Philadelphia sample is higher stress and lower SES than the sample we collected in Boston. If we then look at, okay, now which areas are showing a relationship with income? Positive correlations with income. In Boston, we see just these sort of lateral occipital areas, right? It's not widespread yet. In Philadelphia, we see a similar kind of pattern, lateral occipital areas, not widespread. So this is important for 2 reasons. So one reason is that these cortical thickness differences emerge over time, right? That gives us hope that if we intervene early, we might be able to prevent what I just showed you on the previous slide of much wider spread differences in cortical thickness. The other thing that I think is important about this that I think has been overlooked is that if you were looking for a behavioral marker of this phenomenon, you would look at visual processes and not cognitive processes, right? So if we start thinking about how at scale we could screen for faster maturation, you might want to look at something like binocular rivalry that's sensitive to GABA and V1 instead of some of these prefrontal tasks. And so that is a bit of a controversial claim, but now that I've seen it in 2 different data sets, that this is really about visual cortex in this age range, I am more and more motivated to pursue understanding visual processes in these kids. Okay, let's now switch gears to thinking about functional brain network development. And this first study that I'll show you it was done by Ursula Tooley in collaboration with Danny Bassett and Ted Satterthwaite. And she's looking at clustering coefficient as a way of thinking about network segregation. And to give you an intuition of what this is, This area would have a low clustering coefficient because its neighbors don't talk to each other. So you're not seeing triangles among these areas. This network has a higher clustering coefficient if you focus on this area in particular, right? Its neighbors talk to each other. You can also get a low clustering coefficient if there's sort of massive overconnections. So what she wanted to know is whether the development of this sort of efficient network structure was different for kids from lower SES neighborhoods versus higher SES neighborhoods. And when you look across the whole brain, this is what you find, right? So our kids in higher SES neighborhoods are starting off looking younger, right? So they're starting off with a lower clustering coefficient, which might be due to just lots and lots of connections for those kids. And then they shoot up. And by the time they're adults, they have this sort of more efficient network structure. Whereas for the kids from lower SES neighborhoods, it looks flatter, right? That maybe they matured early and then didn't show big age-related changes throughout adolescence. And then she wanted to know which areas of the brain were actually driving these interaction effects. And what she found is that there was really big effects in motor areas. Right? Again, it's not really something that we would think that there are big motor differences by SES. I think it might have to do with the age range, right? That maybe you're looking at these 10-year-olds where you get that big difference at an age when motor cortex is developing most. Right? So just to map that back out to this model I showed you before with a snapshot at age 10, it may be that motor cortex is showing these big SES effects because that's where we are in its developmental trajectory. One way to think about looking at this is to look at network dynamics in the ABCD age range. And what we find is the only network that shows a systematic effect of both individual SES and neighborhood SES is the somatomotor system. And individuals from higher SES backgrounds look younger, right? Matching this kind of model. Okay. So now I'm going to dramatically shift away from the brain, back out to the body, because we think that body maturation is related to these brain maturation processes. And so we want to know whether lower SES is actually associated with faster maturation of the entire body. And there's been some evidence that this is true. So both lower SES and stress have been associated with earlier puberty onset and earlier epigenetic signs of aging. But what we really want to know is whether we can detect this earlier, right? Waiting until puberty might be too late. And epigenetics are not very scalable right now. It's not an easy measurement. And so what we're looking at instead is actually molar eruption. And we drew this idea from looking at the evolutionary biology literature, which uses molar eruption as a way of marking the length of childhood. Okay, so we thought, okay, if kids are experiencing stress and their childhood is getting shortened and they're growing up too fast, we're going to see that in earlier molar eruption. And we're going to measure molar eruption with our T2-weighted MRI. And so many of you may have the same data. You can check us on this, and we can look at the molar. Right? So at stage 1 in molar eruption, you can see the follicular sac, that's a hard word for me to say, is present. And we go up to stage 2, that sac gets reduced, the tooth moves up to the soft tissues, right? Then the tooth moves up to the oral cavity, and finally it's fully erupted and in occlusion. And so this is how we're measuring it with MRI. And then we can see whether our measurement system is matching clinical norms. And so when we look at the age of the child against their molar maturity, right, we see that goes up in the way we would expect between ages 4 and 7. Right? These are traditionally called the 6-year molars. And we also can see that girls are growing up faster than boys. Right? This is something that is known in the clinical literature as well. A good sort of sanity check here. Next, we wanted to see how molar maturity was related to income. And what we see is kids from higher income backgrounds have much younger looking molars. Kids from lower income backgrounds have greater molar maturity. And this was all pre-registered. And we see a similar pattern if we instead look to ACEs. So kids who've experienced more ACEs have older looking teeth. This is something we've since been able to replicate in the NHANES nationally representative data set, both for these first 6-year molars and also for the 12-year molars. And we can do some additional analyses in this enormous data set to show that it's really not about things like lead exposure, right? What you might expect to lead to some of this allostatic wear and tear. It's not about nutrition. And so we haven't specifically nailed down that this is a stress effect, but we think it's consistent with that interpretation. So to put this all together, it's looking like lower SES is associated with faster maturation, both of the brain and the body. And we have some new ideas for how we might be able to screen for these things. And I'll come back to those ideas towards the end of the talk. And just to bring you back to this sort of animal model framework, the reason that we're worried about this early maturation is the idea that if stress is creating early developmental breaks on plasticity, that's going to be a problem for learning. But we might also want to know if these same experiences are impacting dopamine circuitry, right? Because that might create additional challenges with learning. And so the way we looked at this is looking at ventral tegmental area connectivity. And I will say up front, we don't know for sure this is dopamine, right? There are also top-down projections to the VTA. VTA has non-dopaminergic connections as well, but it's a first pass. And we started our fascination with the VTA based on work by Kate Pena, who's now at Princeton, showing that early life stress reduces OTX2 expression in the VTA, which causes reduced exploration, so both social exploration and novel object exploration in response to adult stress in mice, right? So that's showing that whatever is going on in the VTA is very important for these downstream learning behaviors. And this is particularly interesting because OTX2 plays such a key role in plasticity regulation. And so sort of another pathway by which these early experiences are changing plasticity. But of course, we want to be able to go from this animal work to understanding this in humans. So graduate student Ann Park looked at how early life stress impacted or is associated more specifically with the pace of VTA connectivity development in humans. I'll tell you a little bit more about this sample that we have. So we have 88 kids. They are sort of racially and ethnically representative of our neighborhood in West Philadelphia. This is our income distribution to show that it's skewed towards lower income, and our education distribution to show that sort of the modal amount of education is a high school degree, but we do have some nice spread. We're also going to be looking at ACEs here, just because of their consistent linking to adult health outcomes. And what we see is sort of often seen with ACEs that you have a skew, right? Most kids thankfully have 0, and quite a few have 1, and then you have this long tail. And I just wanted to characterize that for you. So we have sort of the highest incidence of these household dysfunction experiences, mental illness, incarceration, domestic violence, substance abuse, and divorce, and then lower incidence of neglect and abuse. But this is at least double what we've seen in other data sets, nationally representative data sets. Okay, so we're still sort of enriched for these kinds of experiences because it's probably associated with the higher poverty in our sample. Okay. So the first thing that we can check here, because VTA is such a small area, is just that its functional connectivity is tracking with known anatomy. So this is an inflated brain, medial surfaces, lateral surfaces, high connectivity with medial prefrontal cortex, also visual cortex. This is now a mid-sagittal slice. You can see that we're getting the expected brainstem and cerebellar connections as well. And I just wanted to show you an image of what D1 receptor expression should look like from careful anatomy work to show that we're matching up here, right? So with this medial prefrontal connection and also visual areas as well. Okay, so now we're going to ask this question about how SES is related to the developmental trajectory of VTA connectivity. Okay, so this is an SES by age interaction for VTA connectivity. And what we see is an area just in medial prefrontal cortex. And to plot that for you, what you see is that kids from higher SES backgrounds show an increase in connectivity in this area from age 4 to age 9, almost 10. Kids from lower SES backgrounds don't show that same increase. And they show a blunted pattern here. We can also look at ACEs, now controlling for SES. We see a similar region of medial PFC, right, where we see that kids with no ACEs exposure in red show a developmental increase in connectivity here. Kids with 2 or more are flat. So again, we don't know that this is definitely dopamine, but I think it raises some interesting questions about how this trajectory is related to motivation behaviors, how it might be related to plasticity and learning, but also processes like anhedonia, for example. And you can think about what it's like to be one of these kids, right? If you are constantly predicting that things will be better than they end up being, you might see this kind of pattern, right? If things are always good, you might get this experience-dependent connectivity increase between VTA and MPFC. And if things are always bad, you might not see that increase. Okay. So just to sum up, we see this association for both lower SES and ACEs adverse childhood experiences with blunted VTA functional connectivity development. Okay. So in this last section, I want to change gears to thinking about how we might build interventions to increase plasticity, right? How can we build up these motivation processes, right? And these kids who might not be showing these developmental increases or who might be growing up too fast. And we've focused on 2 kinds of motivation processes. We've looked at persistence and curiosity. I'm going to start with persistence. So the first way we can go about this is just to understand variability across kids. So the kids who are very persistent, how are their experiences different than the kids who are not so persistent? And we started by just looking at interactions with parents. So we gave kids puzzles that we knew were too hard for them because they're from the Wechsler intelligence test. So they're normed for older kids. We knew the kids would have never seen them before because you can't just buy them off the shelf. And we just watched. We said, okay, what are parents going to do? Are they going to encourage their kids? How much do they use indirect instruction or pedagogical questions? How much do they just tell kids what to do? When do they tell kids, you know what, this is too hard, just give up? Or when do they reach in and take over and actually place the pieces for their kids? And we asked how these parenting behaviors related to child persistence as measured by parent report. Right? And parent report has some shortcomings, but parents are able to report on their kid, not just in the minute, the way that an assessment in lab might, but sort of how their kid behaves across many different contexts over time. So let me first just show you what parents did, right? So for each of these behaviors, I'm giving you a number of, on average, how often it happened and how these behaviors relate to other behaviors. So indirect instruction, pedagogical questions, encouragement, direct instruction, giving up, or taking over. So taking over happens about 5 times in each of these 5-minute blocks. The positive behaviors relate to each other, right? Giving up and taking over relate to each other, but not significantly. So this sort of pattern of behaviors makes sense, but the real question is how they relate to child persistence. And the only one that is significant here is taking over. So parents who take over more rate their children as less persistent. And this could arise for a number of reasons. So for example, if a parent knows that their kid's not very persistent, they might want to help and take over more. Or parents who are less patient have kids who are less patient. And so what we did is we used this correlational approach to then design an experiment and test if taking over can cause kids to persist less. So this is the experiment. We have 2 conditions. So the taking over condition, where the experimenter reaches in and says, this is hard, let me just do it. We have the teaching condition, which was inspired by sort of the best parenting behaviors that we saw in these videos, where the experimenter carefully and patiently teaches the kid how to do this puzzle without directly doing it for them. And then we have a baseline condition. And after these conditions, we see how long a child will work on a box that is secretly glued shut. So there's something inside, they can hear it, but they don't know how to open it. And then we put this very fun looking toy right next to them and we say, okay, whenever you're done working on this puzzle box, you can play with this toy instead. Okay, so what happened? First, we can look at baseline, right? You just give them the box and you can see that kids will work on it for about 2 minutes. If we patiently teach them, you get no change against baseline. If you instead take over and tell the kid, okay, you can't do this, this is too hard, that you see they persist about half as long. Right? And so taking over is actually causing kids to persist less on a different task. And I found this to be a really deep and actionable insight. So I find myself trying to take over all the time, right? It's really hard to watch people around you struggle. And it is very tempting to just step in and do it for them. It will be faster. They won't have to get frustrated. But we're just starting to think about, okay, what does that mean to the kid? The kid is seeing that you don't think they can do it. And we've tried to undo this effect. We know that sometimes parents have to take over. We know that sometimes situations are dangerous or you're in a huge rush. And there should be ways to reframe it so that you don't have this same effect on motivation, but it's hard to do. So we've tried things like telling a kid that you're going to take turns, right? It's my turn, then your turn. It reduces the effect of taking over a little bit, but not completely. So this is still a work in progress, figuring out how you can take over without demotivating kids. Okay. We can also try to learn from variability and persistence within child. Instead of looking at which kids are persistent and which kids are not so persistent, when are kids having their best days and when are they having their worst days? So let me just give you an example of a kid on a good day.
Good job.
Working on this task.
Super toxic. Other side, honey.
Working really hard for 34 seconds. Here's maybe a not so good day.
I'm a little sleepy. A little longer. Oh, yeah.
Was not going to do it, right? She's not going to work hard on this task on this day. Right? Same kid. And so what we wanted to understand here are like, what are the experiences that explain these daily fluctuations in kids' persistence? So for about 2 weeks, we did daily questions of child sleep, child mood, and parent stress. And we had them send us videos of nightly toothbrushing. I'll show you one more toothbrushing video.
Get all those bad guys out. Keep trying.
Okay, so you can hear some of the. Okay, you can hear him say, "Keep trying." Okay, so you can actually get a pretty rich sense of that parent-child interaction over time. And there's just really not a lot of data looking at within-person variability in parenting and what kids then do. And what we see is that the strongest predictor of how long kids will brush for is parent encouragement. And this is not direct instruction. So if you tell a kid, brush your teeth, it doesn't work especially well, right? It's really this talk and encouragement that seems to be within kid associated with how long they'll brush their teeth. It's true across kids too, really strong within kid. Kids will brush longer when they're in a better mood. Right? It sort of shows you that this method is matching our expectations. It's true for some kids, but not all, that they will brush longer if they slept well the night before. Mm-hmm. And same for parent stress, right? Some kids brush longer when their parents are stressed, some kids brush less, but there's no net effect here. And so across these studies, we can start working to figure out how we can enhance kids' persistence, right? So we can think about interventions for parents and teachers to take over less. We can think about how we can encourage people to encourage more. And then we can work on diagnosing what are the factors that matter for each kid, right? So if we figure out this kid is sensitive to sleep, right? Let's intervene on sleep. If the kid is not sensitive to sleep, that doesn't make any sense at all. Right, so we've sort of developed this tool now that can be used to measure individual kids during an activity that they have to do anyway. That's good for their health, right? That's easy for a parent to send us footage of. And so we're really excited about sort of the different doors that we've opened with this new method. Okay. Next, let's talk a little bit about curiosity. And this is newer work. But here we want to think about how we can boost curiosity, because there's lots of work suggesting that when people are more curious, they have greater VTA activation and greater connectivity between VTA and hippocampus, right? So if we can help kids sort of up-modulate VTA activation, up-modulate dopamine, can we help them learn more? And this is a collaboration with Liz Bonowitz, who's at Harvard Graduate School of Education, her student Joseph Colantonio, and then in my lab, Sophie Sharp, Greer Bizzle-Hatchell, and Christina Recto. And what Bonowitz's previous work showed is that when parents ask questions, their kids will be more curious, they'll explore more. What we wanted to know is if kids ask more questions, right, do they learn more? And then maybe downstream, are they showing increases on other behavioral aspects of curiosity? And this is all very new, but what we've designed is an 8-lesson curriculum where we can look at learning. So we have these short lessons that include a story, a video, and an activity. And we can do random assignment to have kids either be encouraged to ask lots of questions or to listen carefully, which we think is more sort of learning as usual. And we can see at the end, do we see gains in learning? So just to show you some data here, very preliminary, where we've pre-registered this up to in a 55 per condition, so we're less than halfway through analysis. We have kids in the listening condition and kids in the question asking condition. And then at the end of these 8 lessons, we just say, okay, like, here are some questions about science. What do you remember? And we see that the kids in the question asking condition remember significantly more, right? So that practice of asking questions is helping them learn more, which I think is really exciting. And then we can sort of see whether we see transfer to other aspects of curiosity. We can also do really cool analyses within these 8 lessons because they're over Zoom, looking at daily fluctuations in kids' attention, right? How that's relating to fluctuations in their sleep, in their exercise, in their time outdoors, in their parents' stress. So we can sort of apply that toothbrushing, EMA, or ecological momentary assessment approach to looking at these fluctuations in curiosity and these fluctuations in learning. Okay, so across this whole line of work, we think that there's promise to boost motivation by reducing taking over, encouraging encouragement, and perhaps promoting question asking, but this is newer. And that we can start working towards figuring out the factors that matter for individual kids. Okay, in the last few minutes, I want to zoom out again and start thinking about how this line of work can actually help kids learn. Right? I think I started with this problem that during COVID, we're seeing huge learning losses and that we're all really motivated to build education back better, and how might we use some of these scientific advances to do that. So first, I think there's some promise for characterizing these developmental changes in plasticity. Right. So in practice, that might mean something like guiding the timing of interventions, right? Intervening at the inflection point, which might differ across kids versus at very high or very low plasticity. But I've also done some work suggesting that for at least the frontoparietal network, there remains plasticity into adulthood. So when we follow adults as they engage in reasoning training, we see changes in diffusion in these lateral, sort of late developing lateral prefrontal areas and in connections between them. But also interestingly, I sort of went back to notice this about the data, but we see changes in diffusion right around ventral tegmental area as well. Right? This reasoning training was highly motivating. And so we can see that there's actually some malleability even in the brainstem, which I think is fascinating. And then in terms of policy here, we can think about how these kinds of studies can provide support maybe for early intervention if you're thinking about what might shift all of these curves later, right? Versus support for lifelong educational investment, right? Coming back to the idea that plasticity is never zero. We can also think about developing markers of early maturation, right? So I pointed out that we have this evidence now that kids from lower income backgrounds are getting their molars earlier, right? In practice, this means that we can screen for early maturation. I've been talking with my graduate students, Austin Borashak and Cassidy McDermott, who are in the clinical program. Right? And it's very important to them that they can work with individual kids and say, okay, like you are on an accelerated trajectory, right? And right now that means they're hitting puberty earlier, but maybe we can catch them even earlier to say, okay, what does that mean for you as a learner? What does that mean for you in a classroom where you look more mature than the other kids around you? What does that mean for the teachers who are interacting with you and their expectations for you, right? So I think there's going to be some implications for the individual child here, right? As well as ways of coming up with interventions at a broader scale. And in terms of policy, I think this is just additional evidence that kids' experiences get embedded in their bodies. Right? So we have known for a long time that individuals from lower SES backgrounds have higher morbidity, earlier mortality. Right? The fact that we're seeing these kinds of early aging processes when they're 6, I think can be seen as a wake-up call to invest more in making sure that this doesn't happen. Right? And so maybe part of this is going to be tracked to environmental exposures, like air pollution or something that causes inflammatory processes. But I think we're also going to see that it's can be traced to discrimination, to racism, to those sort of early stressors that are persistent and really get under kids' skins. We have to think about what we can do at a policy level to prevent that. And the last thing I want to mention is that we, as neuroscientists, might have some new ideas about how we can intervene. And so I've done some work on reasoning, sort of both in a way that was led by researchers and then led by teachers. And the core of this was to make these games as different and novel as possible, as fun as possible, because we knew from the neuroscience that this motivation piece was absolutely critical for brain plasticity. And so I think we can build some of these kinds of interventions as neuroscientists. And then this can lead to some recommendations for practice. Right? So we can recommend to teachers and parents that games are part of their classroom. And you know, the evidence on this is not ironclad. Right? These kinds of studies are really hard to do. But it's still a recommendation that I give because the cost is pretty low. Right? So telling a parent that they should play games with their kid, it's pretty unlikely that that is going to backfire. When you talk more about how classroom time should be used, that's dicier, right? Because every minute is so precious, and a minute spent playing games is a minute not, say, doing physical exercise, which we think is also really important. But I think we can sort of work our way towards making more specific recommendations for practice here. And then in terms of policy, I think we can advocate for more support for innovation in education. A lot of this research was possible because of partnerships with schools that allowed us to try new things, to sort of work across nonprofits and schools and funders to develop incubation spaces for these new ideas. And then we also have lines of work on just evaluating existing interventions where you give people books and toys when their kids are really little, and figure out how that is going to impact healthy brain development down the road, right? Just to create additional support for those kinds of programs. Okay. Let me close by saying that I've been focused on how the environment is shaping the brain and the mind in early childhood. But I think there's no reason to believe that these exposures and experiences are limited to just kids. And so if I think about all of you, many of you are experiencing social isolation, or you're experiencing grief, or you're experiencing boredom, and that's shaping your brain and it's shaping mine. And I just look forward to a time when I can be giving this talk in person and meet some of you that I don't know, to have that level of cognitive enrichment to lead to better brains and better ideas, hopefully for all of us. So thank you for your attention.
Thank you very much. I have a number of questions, but I see that I was secretly hoping there won't be too many questions, but I do see a lot of great questions here. So let me start and I'll read it. And if you're the author of the question, and if you want to supplement, please do. But do you want to keep your slide up, or would you like to
close them? Yeah.
Other people? So that might be nice. Great. Thank you very much for a wonderful talk again. Let me see if I can read this. All right. From John Hegarty, how do you envision these brain behavior relationships in children with neurodevelopmental disorders in which persistence may be affected, such as restricted and repetitive behaviors in autism? And there'll be another autism question coming up after this.
So it's a really interesting question. I don't do much direct work in autism, but I think that there's a lot of interest in how plasticity might be different, and sort of how motivation systems might be different, what kids are finding rewarding or not. And I get questions from parents quite a lot about whether taking over is going to have the same effects in kids with autism as without. I don't think we know, but I do think a lot of kids with autism still are paying attention to us, and they are still trying to figure out what is worth their effort, when they should persist, and we can use our behaviors to guide them to working harder at the things that matter, where they're likely to have positive results.
Great, thank you. If you want to add, please do chime in, but otherwise we're going to move on to the next question. The next one is from Debbie Fine. Also wondering about autism, specifically seems like intervention before age 2 is more effective than later, but your curves look like plasticity hasn't started to decline at age 2 in any network.
Yes, I think that's really interesting, and I think there's more evidence for early intervention in autism than maybe in typical development. But I think if you're trying to change the shape of these curves, then you need to start as early as possible, right? Versus trying to target behaviors that emerge from specific networks. And so I do think that one sort of idea that falls out of these curves is that you need consistent intervention early, right? If you do something once when a kid is 1, it will get overwritten almost immediately. But if you're able to have consistent intervention and consistently changing their experiences over weeks or months or years, then you're more likely to change sort of slopes of development after that.
Great, thank you. Next question is from Anna Schusterman. In the SES and ACEs analysis, can you put both SES and ACEs in the same analysis? For example, does ACEs score mediate SES effects, and are they separable, for example?
So we try to look at that, and for the VTA functional connectivity results, the SES and the ACEs patterns are almost identical, whether or not you control for the other. Right, so it seems like they're both tapping into similar kinds of mechanisms. In the molar eruption analyses, income is much, much stronger than ACEs. And so that might be something more about like the chronic aspects of poverty. It may be something more about how ACEs are measured and the fact that we have so many zeros there. You have to remember that because they're sort of these young kids, 4 to 7, a lot of the bad things might not have happened to them yet. So you might actually find that ACEs are a better measure later in childhood than earlier.
Great, thank you. Next one is just a comment from your prior mentor, Sylvia Bungay, and she says, such exciting and innovative work. Thanks so much. Catherine Humphreys, amazing work. Thank you for sharing. Tsipi from Israel. She's up still, amazing talk. Let's get to some questions. Tsipi, were you, Tsipi Horowitz-Kraus, question from Israel. Were you able to compare BW, B/W, what does that stand for? I'm not thinking, thank you, between the subgroups of ACEs, or only looking at the comparison of ACEs 1, ACEs 2, and so on? Sorry, my brain is sleeping.
It's a great question from Israel. You might just be tired too. So we haven't been able to look at the individual stressors yet, just as a sample size problem. Oh, you have a cute person with you. Hello, cute person. It's so nice to see you. But it's something that we'd love to do. I think that my working model is that we're using it for a proxy of cumulative stress burden. But certainly if you were to look at individuals, you might see that the genetic risk contribution might look different for substance abuse than it does for mental illness, than it does for divorce. And so that might help us start to think about what the specific experiences are that are modifiable. I love that idea.
Great, thank you. Does anyone else have questions? You can just unmute yourself and ask questions if you'd like. No questions? I'll ask one. I have a number of them, but I'll just ask a question which seems probably like a naive question. But for example, you were talking about differences in kind of accelerated maturation in low SES and certain populations. So is the advantage, I'm just trying to play sort of the devil's advocate here, but is the advantage of using neuroscience over, for example, measuring, since you showed that it correlates with body accelerated maturation, so instead of using puberty as a metric and doing that, is it because it gives you more specific, network specific or function specific acceleration index? Or is there any other added advantage over kind of a blunt measure of puberty or a combination of body maturation sort of index?
Yeah, I think I would love to get to a screener that is molar eruption or is binocular rivalry in young kids because it's scalable in a way that brain imaging is not. I think the advantage of brain imaging is that it is the bridge between these causal models in animals where you can look at actual changes in plasticity to what we can observe in humans. And so part of it is just interest, right? I'm a neuroscientist. I want to understand how these processes work. And with basic science, you never know what the recommendations are that are going to fall out of it, right? I didn't start out being a person who was interested in molars, but sort of discovering that there were cortical thickness differences that look really broad made me think about sort of other ways of thinking about accelerated development. I started out in this career because of you, interested in sort of what neuroscience can add to education. And I think it's still really an open question, right? There's still a lot of promise, but it's still hard to point to a specific finding that has changed practice dramatically.
Great. Thank you very much. Any other final questions?
I had another question that's about policy and enrichment. So you read about these programs for kids living in poverty in cities, and then they get to go to summer camp, once a summer for 2 weeks, and it's a completely different novel environment, and they're away from their stressors. That always seemed to me like so too little, too late, but given your quadrants, maybe it's not.
Yeah, I think it's a really interesting point. And I guess I do think that there's some promise there. And I think that there's some evidence for programs like Outward Bound when you have an experience that is deep and creates that sense of awe, right? That sort of triggers that your knowledge networks are not sufficient to explain everything around you. And so you may get pretty deep belief changes and even changes in these trajectories from brief experiences. And that shows up in memories too, right? Kids might have better memories for their summer vacations than they do for the sort of daily grind of first grade. I agree with you that it probably is too little too late, that there might be sort of a hybrid approach with the investment earlier and more holistically, in addition to these kinds of novel experiences later. Great question.
Great, thank you. One final question that I see here from Leslie Snyder, if it's okay with you. Any additional advice for helping college students, older ages on your curves? And then there's one more that came in and we'll close it there. But if you have any answers to that one.
So helping college students, I think about this a lot because I teach college students. I really focus on sleep And there's evidence that sleep matters a lot for learning in college, and Kano Okano and John Gabriele's lab has a nice paper on this. So I talk with them a lot about sleep, though that has caused stress. We're like, I can't sleep. Like, why did you tell me that I can't sleep? But I do think that that matters. I think now a lot of their social support is different, and trying to figure out how to work with college students to still feel supported, still feel connected is going to be the priority. Right? That is sort of what is developmentally most important at that age. And I think sort of a healthy, happy person is going to be able to learn in your classes better.
Great, thank you. I know we set aside an hour and 15 minutes, so I'm going to ask this last question from Nicholas Camacho, even though I know a lot of people need to go and have left already. The final question is, do you have any ideas on how your work with curiosity might interact with anhedonic presentations of internalizing disorders in children?
Oh, it seems really related. I think that's a great question to try this kind of intervention with kids who are experiencing anhedonia and try to figure out what sparks joy, what sparks interest in these kids. We've designed this curriculum around animals because it is so interesting for so many kids, but you might be able to work with a child to figure out what they're most excited about, even if they're sort of not feeling excited about much, and have them go through the process of trying to come up with questions and trying to find their curiosity. And then once they know how to do that, teach them to extend it to other contexts that they are maybe not so naturally interested in. That's a really important question.
Great, thank you very much. Okay, well, thank you very much for those who stayed over an hour to attend Allyson's talk. I think it was a fantastic, very exciting, innovative talk, and we all learned a lot. Thank you so much, Allyson, and thank you everyone for being here. And I can't unmute today somehow, so I'm not going to let anyone clap, but thank you. So I'm sure everyone's virtually clapping here. Thank you very much.
Thank you, everyone.